share at the moment that before we go to today's main event, which is the Magoun Lecture, and it's not a secret anymore who gives the Magoun Lectureship. Before I do that, it is my enormous pleasure to introduce and welcome this year's Magoun Scholar. Each year, the Neuroscience department is lucky enough to be able to give out 2 prizes. One of them is the Magoun Prize, which we're giving out today, and the other one is the Idison Lectureship. And I already encourage all of you to think about the people you have in your lab, graduate students, postdocs, that might be appropriate for these prizes, and please, nominate them. That's an important part. Today's winner is Jennifer Kong, who is a 6th-year student in the NSIDP program. She came to us from Willamette University in Oregon, where she did some very nice work as an undergraduate, and actually also took the opportunity to go to the place from Oregon, which is sort of closest, also a coast place, which is Hawaii, where she spent 2 summers looking at myelination in invertebrates, which I thought was really very interesting. She then, we had the fortune to attract her here, and Ben Novich had the very great fortune to attract her into his lab to work on notch signaling, patterning in the spinal cord, and I think we can all read more about her on the next slide here. So you can read a little bit on what she's been up to and so on. Oops, there we go. She seems to know that. So it's a very great pleasure to have you, and congratulations. And we have a little plaque for you. We have this, so very nicely framed plaque. There's a little financial award that also goes to you, her, and then actually the only thing that we have for you is that you get to keep Jennifer a little bit longer in your lab. And I give over to Chris Evans.
Thanks, Felix. Congratulations, Jennifer. Okay, so this year's Magoun Lecture is going to be given by Istvan Mody. Istvan's on the right there. This is Tim Magoun, who was very instrumental in starting the Neuroscience Research Institute, UCLA, many years ago. And we thank him for it. Okay, so let's talk a bit about Istvan. He's really, I think, our renowned expert in the electrophysiology and cell biology of GABA receptors. Whenever I think of GABA receptors, I think of Istvan. And I don't think of them much, because I think of other receptors all the time. But when I think of GABA, I do think of Istvan. Istvan got his PhD in the University of British Columbia in Vancouver in Canada. And then he did a postdoc in the Medical Research Council of Canada. And he was the Isaac Walton Killam Memorial Postdoctoral Fellow. Then he went to Stanford. And I think at Stanford we probably overlapped maybe a year or 2. I didn't know you. He was in the Department of Neurology. I think Stanford at that point was very silent, so nobody knew anybody except the neighbors. And Istvan then went on to UT Southwestern, where he was an associate professor of anesthesiology and pain management. And then he came to UCLA in 1995, and presently he's the professor of neurology in the Tony Coelho third chair. So, as it will be clear from the title, Istvan is very interested in inhibition, and GABA is our major inhibitory neurotransmitter. And actually, I've actually read one of his reviews on KCC2, because I think everyone's interested in KCC2, and we became interested in that as well. But I'd like to sort of underline really Istvan's integration into UCLA community and his impact here. And I want to give you a list of the co-authors the co-authored publications he's had with many faculty, and some of these people some of these faculty he's co-authored many different papers. So here's the list. He's collaborated with Giovanni Coppola, Val Carr, Pete Engel, Paul Thompson, Rich Olson, Mike Sofranio, Isan, Tom Carmichael, Alcino Silva. You know, all these are in different areas, right? Caroline Hauser, Jim Bolter on nicotinic receptors, Duran, Felix Schweitzer, and Francisco Benzania, Jack Judy, William Yang, and Julio Braga. So, I mean, there's a whole lot of science which has involved Istvan here, and I'd like to congratulate him on getting this award, and I think it's very well deserved. He's done some great work here, and really has been a very widely collaborated with many people here and contributed to the community. So, very much.
Thank you, Chris. Thank you. Can everybody hear me? Well, thanks very much, and thanks to the community and the nominators and the adjudicators of the prize. I'm really happy. And I don't know if you saw in the first slide that there's only one spot left on the lower right. So I was very happy to kind of slide into that. The other thing I noticed on Chris's slides is how messy my desk is, number one. And also how I probably still have the stereotypes in my lap, if anybody wants to borrow it. So let me switch to my slides now. That I picked this title, watching the wheels go around, and I'm sure you're familiar with the John Lennon song from the lyrics, but basically we have a dysfunctional thing here. Let me see what we can do. I don't have my glasses for that, unfortunately. Yeah, that's not how I envisaged this, though, because, yeah, see, the reason I picked this title is because of, I really love to watch the wheels roll on Inhibition, not because I'm getting off the merry-go-round. That's why that title was there. It really, I want to tell you today about how marvelous this field has become. Because early on, every synapse revolved around excitation, revolved around glutamate synapses. It's only now that we have realized as a neuroscience community that really what I have been preaching all along, that the GABAergic interneurons that provide the diversity in the brain, they're really doing a lot of function, and they are really involved in a lot of diseases of the nervous system, as it will be evident from the snippets that I show you about this. So this is actually a second figure from the Hallmark paper that got Istvan Magoun and Maruzzi the fame that they deserve about the activation of the reticular activating system. And this is in a cat, and what they did, they stimulated basically in the basal forebrain or the reticular activation system in the dulles sometimes and so on, and they saw this very sort of high frequency, low voltage activity that was interrupting the EEG. Now, interestingly enough, if you just hone in, and I did this because I really wondered what that frequency was, and I couldn't count any better, but at this resolution, I counted 38 hertz of the oscillations, and this is particularly in the gamma range, that, as you will see, just now, we know that the neurons that generate this upon stimulation in the basal forebrain are actually GABAergic neurons that project to the cortex, so they're not interneurons, but nonetheless they're GABAergic neurons. And here's the proof. Many years later, this is a paper that just came out in PNAS, that channelrhodopsin activation of the paravalvular interneurons deep inside this basal forebrain, reported them in the cortex, evokes this pretty much the same frequency, 40 Hertz activity that is known to be so important in cognition, attention, and learning and memory, and kind of the binding of various modalities in the brain. So it probably turns out that what Horace Magoun stimulated down here in the cat is actually the same neurons and the effect of the same neurons on there, now that we have these specific probes. I wasn't involved as early on in the stimulating the reticular acetatic formation, but my very first paper that was published in the then famous Canadian Journal of Physiology and Pharmacology, we already speculated that inhibitory interneurons are involved in the effects that we saw on the effect of norepinephrine in increasing excitability in the hippocampus. So this is how far I go back. That was my first paper in graduate school in Vancouver. And this I for back in the notion of interneurons. They're also very beautiful. You have to admit, this is a slide that I borrowed from my friend and colleague Tomáš Freund, who labeled different interneurons in the hippocampus, and then they colored them. They unfortunately don't come in these different colors, although they use various mice now, we can get them to be so different. But look at the high difference in the axonal arbors, for instance. This is a basket cell that innervates just the pyramidal cell layer here. This is an OLM cell that, for instance, just innervates the dendritic arbor, the very tufted dendrites of the pyramidal cell. This cell is a bistratified cell innervating both pyramidal layer and the dendrites. So they're highly specific, and they are really, in the arborization, they innervate thousands of cells being in a position where they can control the activity of several other neurons. And from that comes my sort of Murphy's law of 20-80, or 20% to 80% of the work. In my analogy to this neurons that are GABAergic interneurons being 20% abundance in the brain, and to the switches, the telephone central and the pyramidal cells basically being just the wiring and the telephone post of the system. Of course, now we know that there are different pyramidal cells as well, so there's some diversity within that, but nowhere near as the diversity in the GABAergic system. So much so that I ventured to put on this slide. This is the slide of the Apple stock from actually May 8, 1998 till today. It's 3,020% increase. What happened on May 8, 1998? Anybody know? With Apple, this happened. The iMac was introduced. That's Steve Jobs there, unveiling the iMac. This is how it looked like. I actually had one of those. And too bad I didn't keep it in its box because it would be worth a fortune today. But the I, a lot of people speculated what it stood for, and these were the, given that this is called internet, it's called individual, instruct, inform, inspire. And so this is, if I stands for the internet in the iMac, then I'm sure that iNeuron would be a really good definition for the interneurons, and I very highly suggest to use the logo now on to denote this highly intelligent interneurons in the brain. Now, I talked to you about the diversity, and they represent diversity in the brain. This is a very recent Science article, but they did single-cell RNA-seq to establish different functional varieties of neurons. So actually, in this case, they managed to isolate 16 different types of interneurons in the CA1. This is pyramidal cells in the somatosensory cortex. And these are different layers, obviously, of different types. But in the CA1, there's only about 4 types of pyramidal cells based on RNA-seq. Well, my friend and colleague Peter Mody classifies them differently. And on this slide, it's 23 different kinds of interneurons that are present in the hippocampus. And just to kind of make a point about the cells that contain parvalbumin, because many of us use these viral vectors nowadays, and we have now the 3 animals that express parvalbumin, 3 recombinase in the parvalbumin interneurons, how heterogeneous even such a population is. Here's one parvalbumin cell, that is the exosonic cell, that innervates specifically the axon's initial segment of the pyramidal cell. Another one is the basket cell, a parvalbumin-positive basket cell that only innervates the somata of the pyramidal cells. The other one would be a bistratified cell here, and the other one is the OLM cell that I showed you on the previous slide that innervates the dendritic layer, some of the OLM cells, not all of them, and parvalbumin. So even when we kind of tend to identify these cells by such criteria as parvalbumin content, it turns out that they were very, very diverse at the end. The cells were discovered, these are the basket cells by Golgi, Santiago Cajal, but first of all, Albert von Kerlicka, who called them cord cell, and this is what the basket cell name comes from. And the axoaxonic cell, the other part of these parvalbumin cells, is a Hungarian story pretty much. So I'm pretty proud that János Szentagotay was the one who discovered them. And one of the few mistakes that he's made in his discoveries was that he truly thought for the longest time that these cells are excitatory. Because he managed to show that these cartridges, this is a Chandelier cell, he called them a Chandelier cell, the axoaxonic cell that innervates the axon initial segment, it must be excitatory, so it's the part where the action potential is generated. And it took Ted Jones and Peter Somogyi many years to reverse this and show that these cells are really GABAergic and they actually inhibit the axon initial segment in a part of the cell that is key for its firing the action potential. So this is another, the popularity of interneurons increases and has increased tremendously in the past years. Here's an article where my former colleague Arnie Christine is also involved, this whole group from UCSF that now discuss in this article in Science that the brain's interneurons may have unexpected uses for understanding and treating nervous system disorders, and here is a slide from that paper showing how they envisage this to help neurological and psychiatric disorders. So dissociated neurons of the medial ganglionic eminence, in this case where these cells are growing and multiplying in embryonic stages, or embryonic stem cells could then be used to transplant either striatum, neocortex, hippocampus, or spinal cord. And some of these studies have already been done. And for instance, Baraban has very nice work showing that the transplanted inhibitory interneurons into the hippocampi of epileptic animals totally cures, that tends to cure the disease. So we are very hopeful that these techniques will advance and that we can actually have highly specialized cells being inserted, even more highly specialized than just taking out part of the MGE, but maybe specific cell types of the diverse interneuronal population can be done. Now, I think Chris took sort of a head start on this. I just wanted to show you the collaborators over the years. There's 28 on that list. I don't think Chris managed to get them all. So it was really a fun time being here at UCLA, and it would have not been possible without this really highly neuroscientific community and highly collaborative neuroscientific community. I'm also grateful of being here since when I came here, I received the Tony Coelho Chair in Neurology. Here's Tony doing a recent visit. He used to be a member of Congress, and he was most instrumental in establishing the Americans with Disabilities Act, of which now is the 25 years anniversary, and he will be actually this year named an honorary Loyola Marymount University, and he will get the presidential award as well for that. So I'm really grateful for all his work and his endowment, which enabled us to do many things that probably the NIH would have not supported over the past. So now the scientific part of the talk, I will focus on really 3 different aspects that these 2 have already been published, but just to emphasize the collaborative nature of our studies. And the last one is some unpublished data that I would like to show you. And all 3 of these are really unexpected workings of the interneurons and quote-unquote inhibition in the brain. So the first one was in collaboration with Jim Bolter and my graduate student postdoc and Jim's graduate student. And this is about a very rare but very severe epilepsy. Autosomal dominant nocturnal frontal lobe epilepsy. It's hard to even pronounce. The ADNFLE probably is the better abbreviation for that. It's serious because people wake up in the middle of the night, they have nightmares, affected by the disease, they have nightmares and very severe seizures. And it's also very interesting, and this is why I put this on, because it was a landmark from Jim to generate these mice, and it was a landmark for us to find out what was wrong with the mice. Because this was the very first channelopathy in epilepsy that was ever cloned. And that was cloned in 1995. And much to the surprise of the epilepsy community, it was on a nicotinic acetylcholine receptor, the alpha-4 nicotinic acetylcholine receptor. People who study epilepsy, they never even thought of the nicotinic acetylcholine receptor. And so it turned out that there's some point mutations in this. And then immediately, the nicotinic community jumped onto examining this point mutation. And they described all kinds of things that still didn't explain anything of the epilepsy phenotype that these people had. And basically, gain of function, loss of function, everything was invoked. But until Jim made this mouse that actually, thank God, had seizures, there was no way of even addressing the issue. So this is the mutation. It's in the M2, this membrane lining segment of the alpha-4 subunit, at least. There's some other mutations by now too. I think I'm going to change to the red pointer, because this is petering out. But what Jim generated is the alpha-4 mutant, the SD ortholog of the mouse, the S258F, instead of the S248F, and another alpha-4 that had an anti-leucine. And so when we looked at the mice indeed, as I said, had seizures, and even the heterozygous mice had seizures. Both kinds of mutants had seizures, which was very good because this was supposed to be, after all, an autosomal dominant disease, and that's what we expected to have. There was a video which I will not show you now. But when we recorded from these mice, it was evident that even outside the seizures that had a high sort of delta frequency activity, which the controls did not, and that's probably when this delta range, the very lowest frequency, goes up, there's very little theta and gamma activity, which are sort of replaced by the seizures in the mice. This was the prevailing notion in epilepsy research. Probably many of us still think that this is partially correct. But the normal brain has a nice balance between excitation and inhibition. It's just common to say that in the epileptic brain, excitation is the balance is tilted towards excitation instead of inhibition. This is the famous imbalance between excitation and inhibition. I don't think that things work just that easily and nicely, because when we looked at the slices made from these mice in the cortex, and these are pyramidal sound recordings of EPFC, so these are the events going downwards. Here's this enlargement of that. And when we actually perfused nicotine had very little effect in the wild-type mouse, but also it didn't have much effect. If anything, it inhibited the events in the mutant mouse. But not so in the GABAergic iPSCs. So these are now iPSCs going upward. And this was the first time I've seen such a huge effect in my life, having studied at that point for probably 20 years or so inhibition in the brain. I've never seen a 20-fold increase in inhibitory events that nicotine caused in this mutant mouse. Now here was another puzzle again. How is it that you have high inhibition in the brain, and how is it that this mouse is epileptic? And so after many, many experiments, we came out with a reasonable answer. We thought that in this mouse, what happens is that there are nicotinic receptors on the terminals of the GABAergic neurons. Then these mutant nicotinic receptors are actually more calcium permeable than the wild-type ones. And when the nicotinic receptors get activated in the mutant, there's a much more GABA release that happens. So this so far was good, and that what showed that our data was consistent with it. But how does then it cause seizures? And the way that this can cause seizures we think, this wasn't proven except for some experiments that I will show you, is that in addition to inhibition, the GABAergic system is an exquisite mechanism of synchronization as well. Particularly if you shut down the entire system for a while, is when it comes back from this shutdown of many different neurons firing asynchronously, they will all fire coming back from this inhibited state at the same time, for some time causing a synchrony that can just move on and go and become seizures. And this is why we think this is happening, because when we added the GABA antagonist to stop these synapses, or at least to light these synapses, then what happened, the neurons no longer fired in synchrony because each inhibitory event was much reduced and probably wasn't the same depending on toxin being sort of a use-dependent antagonist. And lo and behold, the seizures after picrotoxin and this delta activity went away. This is not in vivo administration. This is showing the same data. The relative delta power went down from here to here in the mutants, in the other mutants. But what's most amazing to us is that the seizures were totally gone in the presence of picrotoxin, which is a GABA receptor antagonist, which if you give to a normal animal, mind you at a higher dose than we did, it should cause seizures. So this, I think, should solve this unexpected puzzle that indeed in this nicotinic acetylcholine receptor mutant mouse, there is a overactivation of the GABAergic synapses, particularly by acetylcholine. So this is why the seizures are produced in sleep, because of very small awakening periods when the cholinergic system gets activated, activate the system out of the blue like the lightning bolt, and that's when the people end up having these seizures and these massive events. Of course, we have not taken this to the clinic yet. I don't think that anybody in their right mind would start giving picrotoxin to people with epilepsy, but I think it just shows you how key this system is for the synchronization of cells. Now, just to on another topic that Tom, I think, will be talking about tomorrow in his Brain Runs lecture, and in much more detail than I will show it here, I will just show you how reduced glial uptake retards the functional recovery after stroke. And for this, I have to introduce you to the form of GABAergic transmission that we were instrumental in discovering, and this is what's called the volume transmission of tonic inhibition. It started a long time ago with Tom Otis at Stanford, where he was my first graduate student, and now he's moved on to bigger and better things. But basically what we saw there, that there was a tonic current that could not be explained by just the synaptic receptor activation. And so there's many ways that one can generate this. So there's perisynaptic transmission, spillover, in other words, then various intersynaptic, the receptors sitting not even at the synapses, and so on. Caroline Hauser and I, we studied the receptors that are located perisynaptically, specified receptors that are located perisynaptically and are activated in this manner. Lo and behold, that was what, as I show you later, the system that was involved in the post-stroke recovery. And this system is regulated quite well. It's regulated by, the ambient GABA levels are regulated by various mechanisms, the GAP2 and the BGT1 that are uptake systems to the astrocytes. The strophin is a release system from astrocytes of GABA. The glutamatergic system that had also uptake systems into neurons by GAT1, and then the extracellular space having all this GABA around and regulated by these various mechanisms. In the extracellular space, the GABA that lingers around generate this tonic inhibition. The receptors that I will show you on the next slide too. Here there are, as of now, some other receptors participate, but the major players are particularly the alpha-4 beta-delta receptors and the alpha-5 beta-gamma receptors. In various areas of the central nervous system, there are quite a lot of these receptors, such as the thalamus, in the neocortex, in certain layers, the hippocampus, in the dentate gyrus in particular, but also CA1, the cerebellum, the granule cells are loaded with these receptors. And essentially, they're found everywhere. It is depending on the GABA concentration in the extracellular space and the receptors of how they distribute in the various cell types. So the problem in stroke is that there's really no treatment. The only treatment available is about a 4.5-hour time window to administer TPA, tissue plasminogen activator immediately, and that's even just to remove the clot. It really doesn't do anything to recovery at all. It has been estimated that only about 10% of the people, even in the United States, if close enough to an academic neurology center or ER, be administered TPA. So it's a very, very small fraction of the people who get treatment. So the hours to days after the stroke, one can try some neuroprotective strategies, reducing the cytotoxicity. This is what mainly has been tried, or rehabilitation in the chronic stage. And this is why it has been so difficult to do anything in stroke, because here's a paper from some time ago that over 1,000 experimental trials have failed in stroke studies. And I think I highlighted this here, that the results question whether the most efficacious drugs are being selected for strokes in clinical trials, and even preclinical studies. And that's, I think, because these studies went all out to neuroprotection, and not necessarily the plasticity that is required to rebuild the damaged nervous system. And so we argued, Tom and I, and our graduate students and postdocs, that perhaps the brain has only one rabbit in the head that you can pull out when it comes to plasticity. And we hypothesized then that the process that hinders the developmental plasticity or physiological plasticity may also impede the plasticity that is required to rebuild the circuits after stroke or after damage. And this plasticity we knew from various studies, and even our own, that this physiological and developmental plasticity is very, very much dependent on these GABA receptors, and in particular on these tonically active GABA receptors that are activated by the extracellular GABA that is present in the extracellular space. So that's where we started looking first. So we caused stroke in mice by basically a very ingenious way of inducing with the green light a compound that creates stroke in the motor hemisphere. We could evaluate the animals' recovery, motor recovery, but then we also cut slices from these animals, and then in the layer 2 pyramidal cells right next to the injury, we started recording and analyzing our electrophysiological recordings of inhibition and excitation. And long story short, the excitation wasn't altered at all, interestingly enough, but what was altered is the tonic inhibition that we recorded. This is the level of this tonic inhibition here. As you can see this from here, is a GABA blocker that is applied here, so that the difference between the dotted line is the basal tonic inhibition. Already we noticed that it was much, much larger in stroke, right? After stroke. This is already 3 days after stroke. When we applied the GAT1 blocker, the tonic inhibition as expected went up, increased in size, because we just blocked the uptake system that is picking up GABA from the extracellular space into the neurons. This compound here blocks the uptake system that picks up GABA into the glial cells, specifically inhibiting the glial uptake. And this in a control animal further indicating that there's a component that is dependent on the GABA uptake into neurons, another component that is dependent on GABA uptake into glial cells. Not so in stroke. So the very large tonic window was there. We could increase it by blocking the neuronal uptake, but couldn't do anything with the glial uptake blocker. Meaning that perhaps the glial uptake was impaired because the blocker no longer worked, and by occlusion, so to say. Now, again, this is just a summary slide. And what we found is that in vivo, treating with the antagonist of these extrasynaptic GABA receptors, which was a compound that acted on alpha-5 receptors, and we had other evidence to show that it's beneficial to block these receptors extrasynaptically, is really beneficial for recovery from stroke. The other parts of the system, we haven't even tried, and we haven't even come close, because we maybe entice the GAP1 activity, the neuronal uptake, to even take up more GABA, to reduce this GABA in the extracellular space. Maybe we could find some way of restoring the GAP2-3 function in the astrocytes, or inhibiting this dystrophin, the release channel of GABA. We could always reduce the extracellular GABA by increasing the GABA transaminase activities, or the presynaptic GABA-B receptor function that would block here and the GABA-B receptors would block the release of GABA. Inserting GABA sponges that would actually soak up GABA, or decreasing the synthetic enzyme of GABA, the GAP, the glutamic acid decarboxylase, decrease interneuron activity, or again the bestrophin-mediated release. So this was the only strategy that we applied, and it was very successful, but many of these could still be applied, and they haven't been explored to my knowledge, even though this paper we published about 5 years ago, 4 years ago. So to me, it's very interesting that many strategies could actually lead to a much more active and better recovery of the stroke that needs to be explored. One of the strategies that we did that was also published by Albert Barth in my lab was to stimulate the glial cells in channel rhodopsin and Cree animals. So these are GFAP mice that were crossed with the channel rhodopsin TD tomato mice. And here's the stroke, and here the way we're plotting these recovery plots from stroke is by measuring the footfalls that the animal makes on a grid. And the footfalls can be categorized as short and shallow. So this is a grid where the mice walk on, and they will invariably put their feet through the grid. And we can measure that with high-resolution video cameras. We can categorize them, and Albert has written a program to do just this. And what happens immediately that each of the falls goes up after stroke, and then during these 3 days, we have put the animals, and only for about minutes a day, we have stimulated the glial cells, hoping that the glia will do something better and not start picking up the GABA that they did not otherwise. As you can see here, these are the effect limb, and this is the sham control, so the black ones are the sham controls. And some of the falls were really responsive to this glial stimulation. In other words, we managed probably to entice the glia to pick up more GABA by stimulating it by channelopsin. I do not know the mechanism, we haven't even looked into that, but the recovery was much more spectacular than the control animals that were not stimulated, or the sham-stimulated animals. So this also shows you that intervention on the glial front might be very interesting and novel strategy to look for recovery after stroke. So finally, I've come to the last part of my talk, where I want to show you another oddity, if you like, on the GABA system. This is unpublished, it's hot off the press, and I wanted to share with you because I find it fascinating. What we started to look at old age animals because of my interest in interneurons, that a couple of years ago, with a collaborative study with the Gladstone people from UCSF, we found that in Alzheimer's mice, the parvalbumin interneurons, what else, right? The parvalbumin interneurons fail early on during the disease. The sodium channels are failing, and if we reconstituted the sodium channels, or actually increased them in the spark cells, the evolution of the Alzheimer's disease in the mouse model was much, much ameliorated. So this is what got me involved in the Alzheimer's aging field. And one of the first things I wanted to try is how something as common as LTP might work in old mice, and just regular mice, no Alzheimer's mice in this case, just regular old mice. Now, when I mean old, they're 2 years old. So these are mice that people normally don't work with. Our adult mice are about 8 months old, and the old mice are about, as I say, 2 years or even older. Some of them we specially ordered from the National Institute of Aging that can supply some old mice, but some of them we bred ourselves. And I tell you, this is not for the faint-hearted. You can breed animals for that long, particularly not DLM. So. Don't get me. found 3 different kinds of responses. Many people don't report about these responses. We found in some slices no LTP at all. This is the old, is always, it's 2 years old again, adult is about 6 to 8 months old, 8-month-old and adult. Then we found a response that looked like this. This is the classical sort of LTP response, both in adults and old animals. And then we found some LTP responses that looked like this, sort of always rising, and we kind of, designed a whole bunch of tests to show that what's the difference and how we're going to tell that this is rising compared to the steady state LTP and so on. So these were again both old and adult. So what was really the difference then? Well, the difference was that the fraction of the slices that showed one or the other kind of LTP was entirely different. So here's the 9% or 15% that was not significantly different that showed no LTP at all. Here's the 17% that showed this rising LTP, and suddenly a whopping 54% in the old, and 69% that showed this steady state leveling LTP that was only 37% in the old. So we changed the fraction of this rising LTP in the old animals tremendously. And when you just average all the LTP data, is already evidence that that really makes a difference, because when you average, regardless of what you classified it before, this is the adult one, and this is the old LTP in these 2-year-old mice that shows this rising phase, again, averaging all the LTP animals, which was and pretty large ends. So we're confident that this is the case. Now, from, I think, the 80s, right down to some good Wikström and Gustafsson paper that the GABAzine or GABA antagonist enhances long-term potentiation. Not a surprise, because when you put on an antagonist of inhibition, you expect the responses to be bigger, and that's exactly what people found. And in fact, most people now, they put in the blocker not to be involved even with the inhibitory events during LTP studies. They put the blocker in and they just increase magnesium to have a little less excitability. So this is something that we expected in the old mice, but in the old mice, the GABAzine actually blocked the LTP instead of the mice. So much to our surprise, this rising LTP without the drug was blocked by the GABAzine. So this already suggested that there must be a GABAergic component in here somewhere, because, and if is a GABAergic component there, it must be something that is depolarizing or masquerading, if you like, as an EPSV instead of an IPSV, because that's the most parsimonious explanation for this. So indeed, then what we did, or what I should say, Isabella did, she started antitheresing the GABA receptor antagonist by picoline with iodide during the LTP phase of the slice. So here the stars are antiphoretic applications of bipicoline. And just to show you what happened indeed, this is the adult and this is the old. The antiphoresis of bipicoline gradually started every time decrease the responses, indicated that indeed there is a GABAergic component in this rising LTP phase of the old animals. And there's another interesting fact of LTP, that it has to be this type of LTP, when it uses theta burst stimulation, is specific to the synapses that you stimulate. So how can you actually prove this? You end up using 2 stimulating electrodes, and you place them such that the 2 fibers that you stimulate are not the same, or basically don't overlap. The way you do that, if you do a paired pulse facilitation on this pulse, and the paired pulse facilitation on that pulse, and they both should facilitate when the same stimulating electrode is activated. But they should never cross-facilitate. And you test for that, and if they do not cross-facilitate, means that they are really not, you're stimulating 2 different fibers. Statinization of this input doesn't lead to any increase in the other input. So the input 2 pretty much stays the same as it was before the LTP. And that's what we have shown, and this is a classical test for synapse specificity. Not so in the old animal. Even when we made sure that the 2 inputs were totally separated, the theta burst stimulation has caused some potentiation here. We just, as you see, we cannot even see the rising field, but nonetheless, the non-tetanized pathway has been potentiated as well. So the synapse specificity was lost in the old animal. So because of all of this, and many others that I didn't show you here, we started to speculate that in many instances, old age repeats development. And during development, the KCC2, the chloride extruder, the potassium chloride co-transporter, is not developed. And neurons are born with a high chloride inside them, that this mechanism has to extrude in order for GABA to be inhibited. So we thought perhaps again, the opposite happens in old age. So we are born and maybe die with less KCC2. And so what we ended up doing, we tried to mimic in adult animals the effect of reducing KCC2 activity by either blocking it with this compound or by artificially increasing the chloride concentration in the cells by an optogenetic means, and that is the halorhodopsin that is used to silence neurons, but it's really a chloride pump. So if you activate it with yellow light for a long time, you inadvertently will raise chloride inside the neurons. And so therefore, by these two approaches, we can make, and in this case we used the mice that they were expressing the halorhodopsin only in pyramidal cells. And this way we can stimulate with yellow light and cause an increase in chloride just in the pyramidal cell, make GABA excitatory now, and see how does an adult mouse slice behave when we do these two treatments. And so basically the adult mouse slice behaved like an old mouse slice, because in the halorhodopsin stimulation that was prior and during this time, so we are loading the cells with chloride through halorhodopsin, it had this characteristic shape of the old type LTP. And without this stimulation, these animals had normal looking LTP or these lines. The same with the blocker of the KCC2. So when we blocked KCC2 and no longer extruded the chloride from the cell, this had to be also a treatment that was there for some time before we undertook these experiments. It was long enough for the cells to be loaded with chloride in the absence of the KCC2, and so there you had the same type of potentiation that we saw in old animals, even though these were adult slicing. So what is the model that we're proposing now? And this is interesting. Kind of think about it, by no means final. So what we're thinking, here's a dendrite of a CA1 pyramidal cell. Here are the two synapses that presumably we're stimulating separately. Here's the GABAergic fibers, numerous, as you saw on one of my first slides, of how diverse these GABAergic fibers are from different neurons and how abundant they are on the dendrites of pyramidal cells. They innervate spine next, they innervate dendrites, shafts, and so on. And basically what we think is that during the theta burst stimulation, calcium must enter, but the chloride that enters is rapidly pumped out by the KCC2, and therefore GABA is still being inhibitory on the other synaptic terminal. And then LTP therefore occurs only in that synaptic terminal, in the synaptic button, in the spine that has seen the calcium rise. In the case of the old animals, because of the decrease in KCC2 activity, at least that's what we think, and that's what our functional assays now of KCC2 and anatomical assays of KCC2 show. During the theta burst stimulation, the chloride cannot be extruded. Basically, there's already a depolarizing gradient of chloride, and therefore the GABA becomes excitatory during these strains, and it spreads also onto the synapse that wasn't stimulated at all, or wasn't tetanized at all. So the calcium ends up rising because of the depolarization not provided by the GABA receptors themselves, inhibitors, mind you. Not anymore, I think, in this case. This spreads, the calcium signal can spread and have a calcium signal in the very spine that was not stimulated, therefore spreading the potentiation from, instead of a specific synapse, to also another synapse. And it has been shown time and time again that if there's no synapse specificity of LTP, there probably is a lack of learning capacity as well. So according to this scheme, if it turns out to be all correct, then in the old mice at least, there is not a lack of plasticity, but there's an overwhelming plasticity, so much so that it spreads to other synapses, and therefore losing the synapse specificity of this process could lead maybe to learning deficiency. Okay, so I will pretty much close with that, and just to show you that over the years I'm standing on the shoulders of these people who I'm very proud of. I trained 32 postdocs, 19 graduate students, 23 undergraduates, and many of them are in various institutions being professors and chairs, also, although Tom should be wiped on that list. And so I'm very, very proud of them and of my mentorship and training activity, how well all of the folks from the lab have been doing. And I'm really grateful to have such great people in the lab. The people now in the lab are here, and many of them participated in the studies that I show you, or some new and even better studies to summarize and to learn more about GABAergic inhibition in the brain. And I would like to thank you for your attention, but not before I want to close by congratulating yesterday's prize winners. That was the Brain Prize, was announced yesterday. I don't know if you know. Some of these folks are our colleagues and friends. Horace Winchell, David Tang, and Winfried Tang, and Arthur Conner have won the 2015 Brain Prize, which is touted as sort of the Nobel Replacement Prize for Neuroscience. And I was wanting to congratulate them too. So thank you very much.
Just before Istvan answers all the questions, we're going to have a reception in the Gonda as for afterwards. So those who are leaving, please don't eat all the food. Any questions?
That's really interesting results at the end. Perhaps I didn't follow the model though. Is that GABAergic component of plasticity associative, meaning that if you say just have the whole thing in ATP, are you still changing the concentration of the chloride?
That's interesting what you say. Actually, we did that, and Guido just told me with the data today just on that very question. And it turns out that the unstimulated pathway, we don't get LTP and APV by the way, but the unstimulated pathway is still rising.
Only on?
Oh, yeah, only the unstimulated pathway. Yeah.
So that's pretty interesting. So if your calcium is flooding into a spine, then you want it to go through the dendrite, then back into narrow spines. So do you imagine the concentrations will maintain? Do you think there's some other way that concentrations will be maintained?
I don't think that it spreads, the calcium that it spreads, because as I just pointed out, that's one of the evidence that we have, that perhaps it doesn't need to, because the untetanized pathway became potentiated in the old animals in APV, meaning that the GABA release onto that spine that was not tetanized was sufficient to actually depolarize and cause calcium entry into that very spine. So I'm not just implying that the calcium will spread back and forth through the dendritic shaft. So I think that's the more likely mechanism.
So I have a question, Istvan. Are you looking for things which upregulate KCC2?
Actually,
so we can all live longer and learn things?
Actually, funny you should ask. Somebody on my previous slide, my former postdoc, Yves de Koning, and Yves de Koning, they have developed these in Canada now in Montreal, and he leads a very large department where the focus is also on KCC2 for no other reason, for pain control. It turns out from the gating theory of pain, we know that the GABAergic cells in the presynaptic afferent depolarization is also dependent on chloride gradients. And so that's where his interest came from. They developed two compounds that actually increase the function of KCC2 We've given that to old slices, not animals, yeah? And it reverts the LTP to an adult-like LTP.
That's what I was hoping. Okay, we got it.
So you're saying that increased plasticity in the old brain could be the source of memory problems, learning problems?
I didn't get the first part of your question.
There is an increased plasticity in the old brain.
You could regard it that way, yes.
Yes,
Yeah, it's a confounding plasticity in the old.
Yeah, that causes learning problems.
Probably.
Do you also see atrophy of the brain in your old mice, the way we do in humans?
We haven't quite looked at that, but so far, I mean, the slices seem to be very healthy, so there's we don't see any epileptiform activity in mice as opposed to people, where, again, the incidence of epilepsy is a U-shaped curve, high at the beginning of life and high also late in life. We don't see that, but we haven't really looked very, very carefully at any big morphological.
Is it possible that the increased plasticity is a result of the loss of neurons is causing the
I don't think so. We're not at that stage yet, so it hasn't been a major neuronal loss yet, even in 2-year-old mice. We have some mice that are 2.5 years old, so maybe we should look there.
Okay, is everyone ready for the reception? Thanks again.